Madam, our paper title is Toxicological Assessment of Selected Pesticide using Cyprinus Carpio is a bioindicator insight from behavioral, genotoxic, and histopathological biomarkers. And this study evaluated under the current supervision of Dr. Shehzad Ghayur in Laboratory of Zoology, Department of Zoology, Hazara University, Mansehra. If we talk about introduction, so the aquaculture industry plays a crucial role in meeting global proteins demand, advancing sustainable development and supporting livelihoods. However, aquatic ecosystems are increasingly threatened by pollution from agriculture runoff, industrial effluents, and aquaculture practices. Among various contaminants, insecticides such as carbosulfan, pyriproxyfen, and thiamethoxam are of particular concern due to their widespread use and high toxicity to aquatic organisms. Detected in tropical waters at concentration ranging from 0.05 to 15 microgram per liter, these chemicals pose significant ecological and human health risks. Cyprinus carpio, an economically important freshwater species, serves as a sensitive bioindicator of aquatic contamination. Behavioral, genotoxic, and histopathological biomarkers provide valuable insights into the sublethal effects of these toxicants. However, most previous studies have assessed single compound exposure, leaving limited understanding of their combined or synergistic impacts. Therefore, this study aims to evaluate and compare the individual and combined effects of the carbosulfan, pyriproxyfen, and thiamethoxam on behavioral, genotoxic, and histopathological responses of the Cyprinus carpio, contributing to the improved ecological risk assessment and sustainable pesticide management. The next step is methodology. So in methodology, first we have collected fishes from Haripur farm, and then we acclimatized to transfer to fish laboratory to Department of Zoology, Hazara University, Mansehra, and after that we have acclimatized for 15 days, and then after acclimatization we have found LC50 because this is the pesticides are lethal, so we calculated the LC50 value, and on the behalf of LC50 we used this chemicals and apply to the fishes. So we have created 5 groups. The first group is G0, which is control group. The second group is G1, which show the experimental group, first one, and which is treated with the carbosulfan. The third group is G3, which treated with the pyriproxyfen, and third one is treated with the thiamethoxam, and the fourth one group is G5, which is treated with the mixture of these 3 pesticides. After 30 days exposure of these pesticides, then we have analysis, behavioral analysis, or and for genotoxic analysis and histopathological analysis, we have collected blood from the caudal vein of fishes, and then we prepared the slides for the genotoxic analysis and histopathological analysis. In the result section, first we have response of Cyprinus carpio. So we observed erratic swimming. In control group, there is no abnormalities of the behavior of Cyprinus carpio, and the rest of all groups, G1, G2, G3, and G4, showed abnormalities in erratic swimming, apercular activity, air gulping, hypoactivity, loss of equilibrium, schooling behavior, stop feeding, widening of mouth, and mucus secretion. The 0 sign indicate normal, 1 indicate mild, and 2 indicate moderate, and the third one indicates severe. In genotoxicity, after
Can you see the slides?
No, madam.
Let me check the issue. Can you see it now?
Yes, sir. The third biomarker is histopathology. In a histopathological study, we have selected four organs. The first one is gill, the second one is liver, and the third one is brain, and the fourth one is intestine. In gill, we observed different abnormalities such as telangiectasia, epithelial uplifting, fusion of secondary lamellae, secondary gill lamellae, curling of secondary gill lamellae, epithelial hyperplasia, lifting of secondary lamellae. In liver, we observe ecnotic necrosis, karyolysis, blood congestion, lymphocyte infiltration, sinusoidal dilation, degenerated hepatocytes. In brain, we observed vacuolation, ecnotic nuclei, glia cells, degenerated neuron, inflammatory cell infiltration, and the last one is intestine, in which we observe destruction of serosa, shortening of villi, goblet cells, and fusion of villi. Summarized histopathological alteration which show the sign indication. The last step is discussion. The extensive use of pesticide in modern agriculture and their subsequent runoff into freshwater ecosystem continue to pose significant ecological risk. Uh, fish being highly sensitive to pollutants and ecologically relevant bioindicator provide a valuable model for assessing sublethal and chronic effects of contaminants. Behavioral toxicology serves as a primary tool to observe changes in fish exposed to pesticides. During current study, Cyprinus Carpio exposed to displayed numerous anomalous behavior, including rapid swimming, hyperactivity, increase in opercular activity, rapid gulping of air, and loss of equilibrium, mucus secretion, widening of mouth, and disturbed schooling behavior, which are in settlement with the Reda et al. 2025, in which treated with the pendimethalin. The presence of micronuclei and other nuclear abnormalities in cells can be considered a universal indicator of contamination, which can be used to quickly and accurately determine the toxicity or alteration in fish and other aquatic organisms. References are added. In present study, pesticide exposed Cyprinus Carpio shows significantly higher micronuclei frequency and other nuclear abnormalities in erythrocyte as compared to control groups, which are in agreement with the Khatun et al. 2025 reported similar erythrocyte nuclear abnormalities frequency in the Channa punctatus exposed to prophenophores. The detrimental impacts of these pesticides on Cyprinus carpio, as observed in the current study, are further elucidated through the histological examination of gill, liver, brain, and intestinal tissues. Our result in line with the previous findings, where C. grippenus was exposed to glyphosate and diuron, respectively. Also, a similar observation in the gills of Cyprinus melegala treated with the chlorofenpyr, dimethoate, and acetamiprid. Histopathological alteration in fish liver are therefore considered reliable biomarkers for assessing and karyolysis, which are in agreement with the report. Reported in similar alteration in the liver of Channa punctatus exposed to prophenophores. In this study, several histopathological changes were observed in the intestine of pesticide treated with the fish, such as destruction of serosa, shortening of villi, goblet cell formation, and fusion of villi, which are in line with the output, and reported similar alteration in C. garpiensis exposed to atrazine. Although the literature on histopathological analysis of fish brain is limited, there is a well-established correlation between brain morphology and ecological adaptation in teleost fish across different taxa. Intoxicated fish exhibited a range of changes in their brain, such as vacuolation, pyknotic nuclei, glial cells function degenerated. Degenerated neuron and inflammatory cell infiltration, which are in settlement with the Ibrahim et al. Reported similar alteration in brain of the Oreochromus niloticus treated with the hexaflumaran.
So, you don't have any conclusion for your study?
Madam, I have but I did not add in.
Okay, so verbally, can you tell what was the conclusion of this study?
Yes, madam.
Okay, please.
Are you done with your presentation, beta? Mr. Mubarak?
जी मैडम.
जी. Beta, you have used pesticides. Did you show any recommendation about, did you observe any difference between the natural and artificial pesticides effects on the Cyprinus Carpio?
Artificial and natural?
Yes.
Madam,
you have used synthetic pesticides.
Yes, madam. Madam, actually this study
your research was based on synthetic or artificial pesticides, but your recommendation should be which one is the good for the system?
The natural one.
The natural one. And what about the control? Which have the good control? Okay, don't worry. Could you point out any single most new or surprising thing you have discovered?
Madam, before this study, the brain study is limited. So we selected brain as a biomarker because brain work are so limited. So we worked on brain histopathology and the mixture of these pesticides.
Okay, thank you very much. You are the young scientist and you have done your effort. Thank you.
Thank you, madam.